we are now going to generate a PCA plot. A PCA stands for Principal Component Analysis, and it is a dimensionality reduction technique that is used in a lot of data analysis. And in gene expression analysis, PCA can be used to explain the variance in gene expression datasets. So to generate a PCA plot, we are going to first perform a variance stabilizing transformation. Then we are going to use the transform values to generate our PCA plots. So this is how we are going to do it. We will perform the variance stabilizing transformation here. So we will say VSD, then we say VST. So the VST function is the variance stabilizing transformation. So we will do this, and then we will specify the deseq object, which is DDS, and then we say blind equals false. So we will execute this command. And after that, we are going to use the transform values to generate a PCA plot, and that is what we are going to do here. So we will say plot PCA. That is the function we use to plot the PCA. So we say plot PCA. I repeat, this is the function that we are going to use to generate the PCA plot. So we have plot PCA, and then we specify VSD. Then we specify the end group. So the end group here will be equals to, and then we are going to indicate the factors. So we have two factors. So we will say sequencing, and then we have what? Treatments. These are the two factors. So let's execute this command. Okay, so we have our PCA plot. So in this plot, we have two dimensions, which we refer to as the principal component. So we have principal component 1, that is PC1, and we also have PC2. So PC1 explains 59% of the variance, and then PC2 explains 26% of the variance. So let's take a look at what you have. And before I even proceed, let me say that there's a color code here to help you to distinguish between the various samples. You can also annotate the samples, okay, and just to try to maybe add more information. So it's possible to add the sample names or the sample IDs here. So that's something that you can do. Now let's continue. Now let's take a look at the first PC, and that is PC1. PC1 explains 59% of the variance in the data. PC1 is separating the treated from the untreated. So how do we know this? If you take a look at the color code here, we see that this color is for paired and treated. We also have this color for single and treated, and we can see them here. So we have paired and treated. We also have single and treated, and then we also have untreated. So we have this color and then this color, and those ones are here. Okay, so looking at this figure, we see a clear separation between the treated and then untreated. Okay, so that is what this PC1 is doing for us. This also means that there is a pattern in the data that allows us to separate these two groups, i.e. treated and then untreated. Okay, so that is what this plot is showing us. Now let's move on to PC2. PC2 is separating the single end dataset from the paired end dataset. So again, let's take a look at our color scheme here. So we have SC, that's a single end, this, and then this. These are all single end data sets. So we have them here. This is single end, and this is also single end. Okay, so you can see all of them here. And we also have the paired end, which are this and then this. That's what we have here. Okay, so this principal components 2 is separating the single end, which are here, from the paired end. Okay, so that is why it's also explaining variance. So in this case, what we are distinguishing or what we are separating are, or it's the type of sequence we are dealing with. That's single end or paired end. And so these are the factors that we had: sequencing, that is single or paired end, and then treatments, that is what treated and untreated. And the PCA plot we have here has explained that for us, because it has been able to separate these factors, okay, for us. So this explains a lot. Okay, so if you have this, then it helps us to be confident that yes, indeed, the experiments we did was good, the data we had is good, because the data had already been labeled. Okay, so we already know that the samples are what we have: paired and treated, paired and untreated, single and treated, single and untreated. And so having the PCA plots here separating all these samples for us into their various groups gives us a higher statistical power, okay, when it comes to our data. So it's a good thing that we are doing. So that's about it. Now, if you take a look at the Galaxy tutorial, it also points out that there seems to be no hidden effects present in the data. Okay, so when we talk about hidden effects, one hidden effect that we can have is what you call batch effects. Okay, in some cases, we can also say these hidden effects are unwanted variation. Okay, this one I'm saying this, so it has to be taken on a case by case situation. So batch effects, which can be a hidden effect or which can be an unwanted variation, it's something that is possible to correct. Okay, you can correct for this when using deseq. So if you know that you have samples that can influence the results, or that can lead to batch effects, then it's possible to correct for this by including any known bad variables in the design. So that's something that you have to look at. So if you take a look at the deseq online documentation, you will have information and that will help you to correct for batch effect. It's something that I'll probably cover in another tutorial, but for now, let's just proceed. Okay, so that's about it for PCA plots. So let's move on to our next visualization, that is heat map. So let's do that now.